This is a lecture session about the VEXAS syndrome. I'm the chair of this session, Hironori Harada from Tokyo University of Pharmacy and Life Sciences. I'd like to welcome our speaker, Dr. David Beck from New York University Grossman School of Medicine and NYU Langone Health. Dr. Beck is an assistant professor in the Department of Medicine and the Department of Biochemistry and Molecular Pharmacology. His research interests are mechanisms underlying the human genetics disease, genomic discovery in autoimmune inflammatory disease, and characterization of the disorders of vigilation. Dr. Beck is well known in the field of the VEXAS syndromes. Today, Dr. Beck will be talking to us about VEXAS syndromes. Could you start?
Hi, everyone. Thank you so much to the organizers for inviting me here for this talk. I'm sorry I can't be here in person, but I'm really excited to share our work about VEXAS syndrome. I have nothing to disclose. Just a little bit of background about VEXAS syndrome. We identified this disease in 2020, looking at patients with recurrent inflammation, severe, refractory, and looking for genetic drivers. And what we found was a shared driver mutation in UBA1 in men with late onset inflammation. This mutation was found to be somatic. Shown here on the right, we have the mutation present, the mutant alleles in blue, the wild type alleles in red. You can see it's present in the peripheral blood in the hematopoietic stem cells. And if you look at the most mature cells in the periphery, the monocytes and neutrophils are predominantly mutant, and the lymphocytes are predominantly wild type. And this mutation is restricted to the blood, not found in the skin. And so from that original identification of a few patients, we now know a lot more about these UBA1 mutations and how they lead to disease and the phenotypes. So we know that this predominantly presents with disease in men in their 60s. It causes a combination of inflammatory and hematologic manifestations, steroid dependent, and really it's a heterogeneous disease. This is showing some of the clinical manifestations that we see. You can see it affects all systems. And what's unique about this genetic diagnosis is that the UBA1 mutations can lead to clinical phenotypes that overlap with known rheumatic diagnoses, like relapsing polychondritis, polyarteritis nodosa, giant cell arteritis. However, they aren't quite like the larger category of that disease. The patients all share the same phenotype of high fevers, requiring steroids, and have these very subspecific manifestations that meet the criteria for different rheumatic diseases. So that's unique for genetic causes of inflammation. And the other unique part about this genetic cause for inflammation is that patients have a predisposition to hematologic conditions. 30 to 40% have myelodysplastic syndrome diagnosis, myeloproliferative neoplasms are seen, plasma cell dyscrasia, and thrombosis. And all patients have macrocytic anemia basically across the board. And what we've continued to learn is that this disease is more common than we initially expected via our sort of rare disease gene discovery approaches. So we know it's present in 1 in 14,000 men, 1 in 14,000 individuals overall, 1 in 4,000 men over the age of 50. This is in one cohort study in the Geisinger mycodes cohort. The penetrance is 100%, at least in this study, in which all individuals who had UBA1 mutations that were pathogenic had disease. The phenotypes were diverse. And here's just a kind of showing again what I've shown before is that although patients have a shared phenotype, they can have these subspecific rheumatic diagnoses, hematologic diagnoses, pulmonary and cardiac manifestations, and dermatologic manifestations. So this is a more common disease than we would have expected. And at the molecular side, we identified that this mutation is at the methionine 41 predominantly, the disease-causing mutation. And UBA1 has two translationally regulated isoforms. The first is UBA1A, which is nuclear and initiated from methionine 1. And the second is UBA1B, which is cytoplasmic and initiated from methionine 41. And most of the mutations that were originally identified were at methionine 41. We expected that this would lead to a loss of the cytoplasmic isoform. And if you look by Western blotting at patient cells on the right, the red cells, and the controls on the left, you can see that patient cells have a loss of UBA1B and emergence of this new isoform, UBA1C. And this is an isoform just produced within disease that's catalytically impaired. And you can see that there's a loss of polyubiquitination below, and there's an increase in multiple stress markers, the phospho-EIF2 alpha. And so what happens in disease is there's an isoform swap from the normally active UBA1B to the inactive UBA1C in the cytoplasm, leading to a loss of cytoplasmic ubiquitination. So we named this disease VEXAS for the vacuoles E1 enzyme X-linked autoinflammatory somatic syndrome. And from that point, we've been trying to understand more about the disease. So we know that UBA1 mutations lead to VEXAS, and we know that there's many clinical manifestations of the disease, but really there's many outstanding questions about UBA1 and inflammation and MDS. So what determines the heterogeneity of disease? Why do some patients have severe disease and some patients have myelodysplastic syndrome? What drives the inflammation in VEXAS syndrome? And what governs the mechanism of disease? And I'll go into detail on the first two projects and quickly summarize the last one for you today. So we really wanted to understand what drives the heterogeneity of disease. And what we were very interested in figuring out was whether or not the reason why some patients went on to having myelodysplastic syndrome with UBA1 mutations and some didn't was potentially co-occurring mutations. So this is collaborative work with my colleagues at the NIH and Mayo Clinic. And as you can see, we looked at the rate of percentage of clonal hematopoiesis mutations in VEXAS patients. And as you can see, there was more CH mutations in VEXAS versus controls, which makes sense because this is an inflammatory disease. And when we looked at the distribution of clonal hematopoiesis mutations, what you can see is that the main culprits that we all know about, DNMT3A and TET2, were the most common, shown in this plot. But what was surprising is that on the left, we have the patients who have MDS and MPN, and on the right, we have those that don't meet diagnostic criteria for that. There is no enrichment for CH mutations in patients with MDS versus without MDS. So it's not that the secondary mutation leads to myelodysplastic syndrome. We did look at risk factors within this cohort of over 100 individuals for the risk factors for mortality. And here you can see CH mutations like DNMT3A or TET2 alone, or DNMT3A hotspot mutation led to the highest risk in terms of a hazard ratio for mortality. So even though these CH mutations do not confer more of a risk for MDS, they do increase mortality risk. And we think this is through inflammation. So together, this leads to worse phenotype for patients. But we have the question of which really is the driver for this disease. And this is something that we're currently trying to work out in models. But the fact that UBA1 mutations occur on their own is clear evidence that UBA1 is a driver on its own. But additionally, we wanted to look at the sequence of events when the UBA1 mutation occurs compared to the CH mutation. And as you can see, we did single cell DNA sequencing. On the left, you can see the different clones. And what's shown here is an individual that had a TET2 mutation that occurred first, and then later a subsequent UBA1 mutation. And the UBA1 TET2 clone is much larger than the TET2 alone clone. A similar situation, but inverted, in which the UBA1 mutation occurs first, and then CH mutations occur down the line. You see that the UBA1 mutation clone is dominant. So regardless of the order of operations, at least in our small cohort, it seems like when UBA1 mutations occur, it takes over in terms of the clonal biology in the bone marrow. And we think that this is important for establishing that UBA1 is a driver for disease and also probably how to treat these individuals going forward. And so we need more longitudinal studies to understand the dynamics of UBA1 in TET2 and DNMT3A, but at least this is evidence that UBA1 is likely the driver of a lot of the co-occurring phenotypes. So I want to switch gears and talk a little bit about inflammation in VEXAS syndrome. And I think the issue is inflammation is one of the most prominent clinical manifestations of patients with VEXAS. It's really not clear why and how loss of an important ubiquitination enzyme leads to inflammation. So our initial studies in zebrafish showed that many different pathways are activated. We're actually able to show that loss of all UBA1 isoforms in blue versus loss of just the cytoplasmic isoform in green led to inflammation in both cases, whereas loss of the nuclear isoform in purple did not. So we could recapitulate the disease phenotype. Loss of UBA1 led to inflammatory pathways. But really, patients seem to have all inflammatory pathways elevated to some extent. So by bulk RNA-seq, by cytokine studies, we've seen many different pathways elevated. And so that doesn't really answer what is the initial cause of inflammation in VEXAS and potentially what are the targetable pathways. So to answer this question, we use single-cell RNA sequencing. And this is just the study design that we used in which we took bone marrow samples from healthy donors and VEXAS patients. We had a cohort of 9 individuals. We looked at bone marrow mononuclear cells and HSPCs and performed a series of assays, including single-cell RNA-seq and flow cytometry profiling to evaluate the different inflammatory pathways involved in disease. And I just want to show you on the right here some of the predicament associated with VEXAS, where if you look at the colony formation assays with bone marrow mononuclear cells, the VEXAS versus controls, the VEXAS patients don't form any colonies. So these cells are really failing to grow in vitro, yet in vivo, they have a clonal advantage. So this is our bone marrow mononuclear cells. And by single-cell RNA-seq, you can see the different populations that we were able to identify. And in the center, what I want to highlight here is that the myeloid cells are predominant in VEXAS. And this is shown on the right in terms of the bar graph, in which there's an increase in terms of the myeloid to lymphoid ratio. So with the UBA1 mutation, there's an increase in the myeloid populations. When we look at the bone marrow mononuclear cells in terms of inflammatory pathways and activated pathways in the cells, what we see is that many different inflammatory pathways are elevated. Interferon gamma, alpha, TNF alpha. But what's striking is that they're not just elevated within the neutrophils and monocytes, the innate immune cells, but we also see them elevated within the adaptive immune system, which are predominantly wild type, suggesting that there may be some non-cell autonomous component to the disease. We also can look at this at the HSC, HSPC level, and the progenitor levels shown in the bottom here at the specific pathways. And what you can see is that the myeloid lineages are more inflamed than the lymphoid, but still there's quite a bit of inflammation occurring in these different lymphoid lineages. We can recapitulate these results from single-cell RNA-seq and identify that the VEXAS cells make more TNF alpha, shown on the right in terms of these ELE spots, the left on these ELE spots, and on the right we have the quantification of these ELE spot assays in which there's more TNF alpha and interferon gamma production from VEXAS cells. So this is kind of confirming what we're seeing by single-cell RNA-seq. We can do similar analyses with the HSPCs, the earliest progenitors in the bone marrow. And again, we're seeing that many different pathways are activated within the granulocyte and the megakaryocyte progenitor lineages. But what's striking is we also see again that the inflammation is present in the lymphoid progenitors. So it's not restricted to the myeloid progenitors. And additionally, we see inflammation in the earliest HSCs. So we're seeing that inflammation is originating at the beginning of where the mutation occurs, which was surprising. And when we compare VEXAS to other related bone marrow failure diseases, whether MDS or CMML, what you can see is that there's an increase in terms of the gene module expression scores for TNF alpha and interferon gamma as compared to in VEXAS as compared to other bone marrow disease. And other than the inflammation that we've kind of identified through other mechanisms, we were able to find this unfolded protein response that's activated within VEXAS versus controls. This was most prominently elevated within the HSCs and the granulocyte and the megakaryocyte progenitors, and less so in the lymphoid progenitors. And this makes sense because what we find is that in UBA1 mutant cells, as I showed earlier, there's activation of multiple stress pathways because there's failure to clear proteins that should be degraded by the ubiquitination system. So this is highlighting a potential mechanism leading to inflammation before the inflammation occurs, because the unfolded protein response is linked to production of cytokines. And so we've been able to identify that inflammation occurs in different lineages and that the different pathways that are activated. We can also, because of the 5' sequencing that's occurring in this single cell RNA-seq, actually identify a mutant from wild type cells. So on the left, you can see the wild type and mutant are labeled wild type blue, mutant red in the bone marrow mononuclear cells, and on the right, the HSPCs. And of course, the lymphoid progenitors, the precursors, are predominantly wild type, whereas the myeloid lineages are predominantly mutant. And so now instead of doing the comparison between control and VEXAS, we can do comparison between wild type and mutant. And what we see is that the mutant cells are highly inflamed in VEXAS compared to wild type cells. So again, similar pathways to what we've seen before, interferon gamma, TNF alpha, and interferon alpha. And again, this is occurring in the HSPCs. And we can actually recapitulate these findings that we've seen consistently at the level of the bone marrow mononuclear cells, HSPCs, and the mutant versus wild type. We can recapitulate this with shRNA knockdown experiments in U937 and THP1 cells, in which loss of UBA1 causes activation of similar pathways. And so I just want to highlight here that we can also identify what's happening in terms of the mutation percentage over time in the different lineages. So this is looking at the ratio of mutant to wild type within a lineage over time, starting from the HSC and moving to lymphoid progenitors, grands, or megakaryocytes. And what you can see is that in the going into the myeloid erythroid lineages, you can see that this is relatively stable, the ratio of myeloid of the mutant to wild type. Whereas when we look at the lymphoid progenitors, there's a gradual drop off in terms of the mutant levels, suggesting that there isn't exactly a break in terms of differentiation of the mutant lymphoid progenitors, but there's a loss, a steady loss over time, that there's a fitness disadvantage to the mutant. So just to summarize the major parts of this talk, VEXAS bone marrow mononuclears and HSPCs are highly inflamed with multiple different pathways. The myeloid lineage predominance in HSPCs is present. We see the UPR pathways activated in VEXAS. We have mutant cells drive inflammations while the wild type cells participate. And I just want to end by talking about some of the more recent findings that we've had in terms of genetic mechanisms and the role of ubiquitination in disease. And this really comes from some of the work from the MLL group, which has shown that in myeloid malignancies and lymphoid malignancies, there's the presence of the VEXAS mutations, the M41 mutations, but there's also this plethora of other mutations that are found, and again in lymphoid malignancies also. So our question is whether or not there are other mutations that may drive VEXAS or this sort of mixed inflammatory bone marrow failure disease. And I just want to refresh you about what UBA1 does. So the ubiquitination system works in 3 steps. There's the E1 enzymes that activate ubiquitin. They work together with 40 E2s that then transfer ubiquitin to 600 substrate-specific E3s. And these are the enzymes that really place ubiquitin on specific pathways. And they target substrates, thousands of substrates for degradation or changing signaling pathways. So really ubiquitination is regulating most cellular processes at any one time. And UBA1 is the major ubiquitin E1 enzyme initiating this whole process. So it's the apex of regulating thousands of proteins, and UBA1 is responsible for over 99% of the E1 activation. So our question was whether or not the MEF41 mutation is unique. Are there other mutations that can cause disease? And this is a collaborative study that we performed with Hakeem Warner from the NIH. We took 183 VEXAS cases, really similar phenotypes to our core VEXAS and 41 mutant patients, from exome sequencing. We've identified novel candidate genes, but today I'm going to talk about the 6 novel recurrent variants in UBA1, and these are listed here. And we wanted to see if these had a similar effect in vitro and in vivo to UBA1 canonical mutations. And this is just to show you the clinical phenotype of these individuals that we identified. You can see that they all have elevated inflammatory markers. They all have anemia. Most of them have fevers and some combination of skin, lung, and cartilage inflammation. And then the majority have MDS diagnosis and are glucocorticoid dependent. So they really have the VEXAS phenotype. And to start to get at the question of if these mutations are functionally impaired, we used a cell line that was generated in some of the Nobel laureate work, initially characterizing ubiquitination, which is a temperature-sensitive Chinese hamster ovary cell line. When you move these cells to 39.5 degrees, UBA1 is lost and ubiquitination is lost. And this is just to show this here, in which you can see in the first lane, there's in the parental CHO cells, there's ubiquitination and UBA1 present. When we heat shock the cells, there's loss of UBA1 and a loss of ubiquitination. And when we rescue with wild type UBA1, we now rescue this ubiquitination effect. And we can test all these mutations using this assay and show that the different mutations, the M41V mutation cause a defect in terms of in vivo polyubiquitination. And similarly, all of the different potential disease-causing mutations cause a defect in terms of polyubiquitination. And this is nice because this is using all the different steps in the ubiquitination process as opposed to in vitro where we would just have to add 1 or 2 steps at once as opposed to the whole cellular milieu. So we next want to see if any of these mutations cause a difference in terms of isoform expression. And on the left, you can see that M41V produces the new isoform UBA1C, whereas all of the other mutations do not cause the isoform swap. So UBA1C expression is likely not required for VEXAS syndrome. And as you can see, UBA1C has a defect in terms of the charging of the E1 enzyme, the first step in this cascade, whereas many of the other mutations do not have a defect at this step. But all of the mutations have defects at the E2, the second step of the ubiquitination pathway, suggesting that this might be the shared mechanism of why UBA1 mutations lead to disease. And so I just want to summarize this part and say that UBA1C is not required for VEXAS syndrome, that there's different mutations that we've identified are defective in thioester activation, but all of them, and we have these unique intramolecular UBA1 mutations that lead to autoubiquitination. But all of the potential VEXAS mutations, all of the disease-causing mutations that have been identified and characterized to date cause a loss of E2 transfer. So that seems to be the bottleneck causing disease in VEXAS. And we're currently working on which E2s are affected more prominently and how this leads to inflammation in my lab, both in cellular and mouse models. So just to conclude this part, I already mentioned that UBA1 mutation are a driver of clonal cytopenia, that UBA1 and CH mutations confer worse survival, and that the mutant cells drive inflammation of VEXAS syndrome and are dominant. And we have these novel mutations that highlight the importance of the E1, E2 binding and charging in disease. And we're currently working on the disease pathogenesis in my laboratory. And with that, before I thank you and take questions, I would like to just highlight the International VEXAS Working Group. There are colleagues from Japan and all over the world involved in this. It's really spearheaded by wonderful French team that have organized clinicians and research together to help provide guidelines clinically to help patients with these disease because it's so new and patients can be quite ill. And with that, I'd like to thank you all for your attention and thank the people that did the work, both in my lab and collaborators at the NIH and Geisinger. And for any questions that you have, I love to get them. Just please email me, and I hope we get to interact sometime soon in person. Thank you very much.
Dr. Beck, thank you very much for your great presentation. Unfortunately, we don't have a great question times for this presentation because time is limited and we have to go to special lunch. So I'm a little bit hungry. So I'm sorry about that. So time is a closed session. Thank you very much.